Hello, this is Benjamin Wolf. I'm an associate professor at Tufts University, and I've been working with the other members of this team to study the microbiology of artisan cheeses in Vermont. And this is all part of the sensory-directed research on artisan cheese to benefit the local, regional, and national dairy industry project that was funded by the Dairy Business Innovation Center for the USDA. And so today I'll be talking about what we've learned from our microbiology research as part of this research project. Of course, the cheeses that we're working with, as you heard from other videos in these webinars, are surface ripened cheeses. And these are cheeses that have a microbial community growing on their surface. That's where the name surface ripened comes from. So this isn't your plastic wrapped cheddar that you would buy at the supermarket. These cheeses have unique microbial communities that define the surfaces and define the aesthetics and define the flavors of these cheeses. These include everything from Limburger to cloth-bound cheddars to Tome de Savoie to a lot of these wrinkly and interesting looking goat cheeses that you sometimes see. The microbiomes of these cheeses, these surface ripened cheeses, include the microbiome of both the paste as well as the rind. And I'll note that these different colors I'm using for microbial cells here are not to indicate the actual colors that the microbes are. For example, the paste of cheese is not green. These are just to indicate different types of microbes. So when you make the cheese, both raw milk and also pasteurized milk can be used. You use rennet and you use starter cultures. There's an initial phase of fermentation where the milk solidifies and becomes curd. You add salt, you age it, and eventually with surface ripened cheeses, you grow a microbial community on the surface and you grow a microbial community in the paste. That on the surface includes both yeast, filamentous fungi or molds, and bacteria. And in the paste is primarily what we call lactic acid bacteria, the same bacteria that we'd find in things like yogurt. And there's lots of different types of these cheeses and different types of microbial communities that form on these cheeses. So for example, in bloomy rind cheeses, you have this sort of white layer of fuzz. That's a mold called Penicillium camemberti. You also have bacteria that can grow there. The two types of cheeses that we've been studying in our project are washed rind cheeses and bloomy rind cheeses, but also to some extent, there's some natural rind-like cheeses involved in this project as well. And each of these different types of cheeses are aged in different ways, managed in different ways, and end up having different microbial communities. Why should we care about the microbial communities on these cheeses? Why is it important to understand their microbiomes or their communities of microbes? Well, one is for flavor development. The microbial communities on these cheeses really drive a lot of the flavors that we perceive in artisan cheese. Because of that surface microbial community, they're slowly decomposing the cheese by releasing enzymes into the curd. That's what's represented here by this sort of spaghetti layer. That would be the rind. And the rind is releasing these little molecular scissors that go in and chop up the proteins in the cheese. And that decomposition process is what releases various flavors that we perceive as interesting or delicious. And that's something that Roy and his team have been studying as part of this project. And some of the compounds that we sense, and these will be things that you'll hear from Scott Frost, who's a chemist as part of this project as well. Some of these compounds are things that kind of smell like cooked cabbage, or might smell meaty, or they may smell buttery, or even like sweaty socks. So there's lots of different ways that as microbes break down the proteins and fats and cheeses, they can release flavor. So a lot of flavor is coming from the microbiome. And different microbes can produce different flavors. Another reason besides flavor development is aesthetics. So each of these cheeses, in addition to having unique flavor development, they also look differently, and that's all because of the microbes. And one reason to think about artisan cheese microbiomes is that sometimes things don't go as planned. So this is an example of a washed rind cheese that sort of has a typical orangey rind, and this is what we'd expect. In this particular case, a particular cheese that we were studying, the cheese rind stopped developing and it started turning purple. It both looked weird, but it also smelled and tasted really bad. So understanding what the microbiomes are and how they shift if there's some kind of major disruption is really important for maintaining the quality of cheese. So how do we measure microbiome diversity in cheese rinds? And I'm going to talk about two kinds of data, so I wanted to explain that. One is a culture-dependent approach. So this depends on growing different types of microbes using agro-petri dishes, like you typically think of when you think of a microbiology lab. A second approach, and this is sort of the more modern microbiology approach, is called culture independent, where you don't actually grow the microbes. What you do is you directly extract DNA or other types of genetic information from the microbe, and you use that information to identify different microbes, just like crime scene investigators might use DNA sequencing to identify who was at a crime scene based on blood or other types of evidence left behind. So measuring microbial diversity using the culture dependent approach involves taking a sample here, it's actually sauerkraut, but this could be cheese, mixing it up in a bag, diluting it out in a way in the lab so that you can spread it across an agar petri dish that has nutrients in it for the microbes to grow. And then eventually, when you look at that after a couple of days, microbial colonies will grow up. And those colonies will have distinct features depending on the type of microbe that you have. So for example, on these agar plates, we were able to selectively grow bacteria from cheese rind. And you can see some of them are lighter in color, some of them are green, some of them are smaller, and those can indicate different types of microbes. On these plates where we were able to selectively isolate fungi from the same sample, you can see that there's some fuzzy little blobs here. Those are molds, specifically penicillium. And these smaller, rounder, shiny colonies are yeast that we found on this cheese. So the culture-dependent approach can be useful. It's actually quite relatively inexpensive, relatively fast, and relatively accessible. In some places that make cheese can even do this in-house in a small, low-tech microbiology lab. The more modern approach, the culture-independent approach, is a little bit more expensive, more time-consuming, and requires a bit more equipment and expertise. And the way that this works is you would have a sample of microbial cells, you'd extract DNA from those cells, and then you can do a couple of different things to that DNA. One is to go in and look for particular fingerprint genes that can tell you what microbes are what, or you could just sequence all of the DNA using an approach called shotgun metagenomics, which is what we did in this project. You then sequence that DNA on a DNA sequencer, the same types of sequencers and platforms that are used for human DNA sequencing. And then what you can do is take that DNA, match it to databases of different microbes, and you get a metagenomic profile. It's called metagenomic because it includes the genomes of multiple different things at the same time, hence the meta. And so here we can see that we have 1, 2, 3, 4, 5 different types of microbes and their names based on this approach. So here's an example of previous data that we collected in a study that I was involved with when I was working with Rachel Dutton. Here, we were looking at both data for bacteria and fungi using that first approach when we went in and looked at particular sequences. And I'll tell you, this is just for the rind, so not including the paste. And what you'll see, these different columns here represent individual cheese rinds that we sampled around the world. So it's sort of like a big global portrait of the types of microbes you can expect to see on cheeses. And you'll see that there's sort of varying amounts of different colors that come and go as you look across this data set. So for example, these cheeses over here have a lot of these sort of red and orange colors, which represent various proteobacteria. Whereas these cheeses over here have a bit more of the green and blue colors, and those represent things like coagulase negative Staphylococcus species, as well as various actinobacteria. You'll also see that similar kind of variation for the fungi. And so this is sort of old baseline data that we have across a broad sampling of cheeses. But what we don't have in this data set is repeated sampling of the same cheese over time to understand how that varies over time. Nor do we have comparisons from the same cheeses of both the rind and the paste. Maybe there's a lot of variation and a lot of diversity in those lactic acid bacteria in the paste. So some of our goals for this project to study these Vermont artisan cheeses were to do the following. One was to understand the microbial diversity of the paste and rind of these 6 cheeses made in New England. Just to understand what is the paste diversity, what is the rind diversity, and how is that structured across these different cheeses. And these are cheeses that are made in 2 different places with different processes. So we wanted to understand how that affected the microbiomes. 2, we want to ask this question, what is more variable? Is it the paste or the rind? So again, most of our previous data is from the rind. We don't really know a lot about the paste microbial diversity. And 3, we want to understand for one cheese, which we selected from one producer, how variable is the microbiome over time if we go back and sample it over and over and over again. And so here are the data that we collected. This is really the sort of meat and potatoes. I guess it's really the cheese and potatoes of our particular study. What I'm showing you on the top are data for the rinds. What I'm showing you on the bottom here are data for the paste. So remember, this is the lactic acid bacteria. This is the really like the curd, the big part of the cheese. And this is just that surface layer, both which play important roles in flavor formation. When you look at these graphs, each of the different colors represents different taxa of various kinds of microbes. And again, each of the different columns here represents a replicate of a sample that we took throughout this study. You'll notice for the cheese named Harbison, we have quite a few bars here because they were taken at different times. So like these 2 samples were taken at one time. This was early in 2022. These samples are taken in the middle, and these are taken more towards later in the year. For many of the other cheeses, we have a lot fewer replicates, but sometimes we actually also have repeated sampling as well. So a couple of things to notice. One is that there is a lot of variation across the different types of cheeses, but not as much maybe as you saw in that earlier figure I showed you. So for example, Harbison, this cheese that we're looking at, looks quite different than a lot of these other cheeses that we studied. So for example, you see all this kind of like bright red coming in and out. Those are things like Vibrio, Stechobacter, and Pseudo-Ultramonas. Those are bacteria that we're able to see kind of come and go in Harbison that we don't typically see in a lot of the other cheeses. A lot of the other cheeses like Ashbrook and Reading, Springbrook Farm, those have a lot more of this bacterium called Corynebacterium, which is an actinobacterium, as well as Brachybacterium and Brevibacterium and Arthropacter. We see those in Ashbrook and Reading. We also see them in the cheese Whitney and Alpha Tolman. Those are made by Jasper Hill. So you can see that each cheese sort of has its unique microbial signature. You'll also notice from the sequencing that the amount of fungi versus bacteria in the rind varies quite a bit. So the fungi, I'll note, are sort of these brown and earthy colors at the top here. So everything from Geotricum up, those are fungi. And you'll see that those are relatively low in abundance in most of the washed rind cheeses. There's a little bit of both Fusarium as well as some of the Deberomyces in Ashbrook and Reading. And we see a lot less of that in Whitney and Alpha Tolman. What's interesting is there's this fungus Penicillium. We see a lot more of that in Harbison. And that's because Harbison is inoculated with that white mold, Penicillium camemberti. So we see that relatively speaking, there can be a lot more fungus in these samples compared to bacteria, compared to these cheeses. All of these cheeses over here are called washed rind cheeses. Those washed rind cheeses tend to not have as much fungal growth on them because the washing process itself breaks up the filamentous growth of fungi, breaks up the mold, and tends to favor bacteria. So it makes sense that we're seeing what we're seeing. One other interesting thing that I noticed is that when we look at these samples here, which were taken in March, and these samples here that were taken in August, again, March and August, we don't see a huge amount of variation temporally for Ashbrook and Reading. Same thing is true for Whitney. We don't see that same kind of variation that we see across Harbison over time. So these are the August samples, and these are some of the March samples. So that's sort of interesting to me that we can see a lot of variation in Harbison. What's going on in the paste? When we look down here, what I think is very interesting is that the paste is kind of boring at the level of taxonomy that we're looking at. It's not boring and that it's not important. It's just not as variable. So you'll notice the colors that we have here. These are lactic acid bacteria, and they're the dominant things in the cheese paste. And that includes things like Lactobacillus species, Streptococcus species, and Lactococcus species. You'll see that we don't have a lot of variation in those. Now, I will point out that in this study, all of these different names that we have here, this is at the genus level. And if you remember from high school biology, you can have genus and then below genus is species, right? So we're Homo sapiens. The sapiens part indicates our species, but there's other Homo species that have occurred, like Homo erectus and other previous ancestors of ours. Just like red oak is different from white oak, which is different from swamp oak. So we're kind of not going to that level here. So there's another level of diversity we may be overlooking. But at this taxonomic level, the rind up here is much more variable than the paste. We do see that in the harbison cheese, we're getting some other things popping up in the paste. Things like, for example, here in green, leuconostoc. We see a little bit of that in Whitney. We never really see any of those over here. Leuconostocs are often found in these sort of softer, younger cheeses like harbison. So that's not surprising to see that. And we also see there's a blip of this thing right here called hafnia coming in and out. And we also have hafnia sort of sometimes thought of as also being common in the rind of cheeses. But in this case, we see hafnia in the paste of harbison, and we don't really see much of it in any of the other cheeses. Okay, so take-home message here is that there's a lot more diversity at the genus level in the rinds of the cheeses that we studied compared to the paste. This could be important when you're thinking about managing these products, where you might be getting variation. It could be potentially variation in the rind. This sort of makes sense. The rind is the part of the cheese that's open and exposed to the aging environment. And while it can be inoculated with particular cultures and managed, this is where things can come and go in the aging environment. New microbes can come in, or microbes can go extinct over time, and you might have a lot of variation there. Whereas the paste is heavily inoculated, usually with lactic acid bacteria in the sort of closed environment of the fermentation vat, and then it's sort of locked into the curd in that anaerobic environment. And so we wouldn't necessarily expect to see a huge amount of variation here. Some of you may be asking, what's up with the little thing called viruses then? Why are there little blips of viruses? Aren't viruses bad? Well, these little blips of viruses that you're seeing come and go, those viruses could actually be what are called bacteriophages, or just commonly phages. Phages are viruses of bacteria. They're not a harm for you and I. They're a harm for bacteria, though. And so these little blips that we're seeing could be examples where viruses are actually attacking the bacteria in the cheese. We don't know that for sure, but work by Rachel Dutton's lab has been showing that there is some interesting phage bacteria interactions that are happening. Great. Another question you may ask is, any of this diversity that we observed different or surprising? And the answer is no, actually. All these various taxa that we're seeing are things that we've seen in cheeses before and things that we would expect to see in these cheeses. So for example, things like this Yaniella we found in our previous study, and we see that popping up here really nicely in alpha Tolman. Yaniella likes high salt of sort of Alpine style cheeses, so it makes sense that we see it in alpha Tolman. You see Staphylococcus, and maybe some of you are thinking, oh no, staph, that's bad, because you think of Staph aureus, but actually in cheese, Staphylococcus is very common as things like Staphylococcus aureum, which is a coagulase negative staph species that's totally safe, and it's not known to cause infections in humans. So yeah, this is the diversity that we would actually expect to find. When we look at this data sort of at a broader sense, using an approach called ordination, sort of separating out the cheeses. So essentially taking this data, making a little dot for each of the samples, and putting them into a broader space to say, how similar are they in composition? Not surprisingly, when we look at harbison, it's all over the place because that was one we have the most samples for, and also is very different from these other cheeses over here, which includes Reading, Ashbrook, Whitney, Tarentaise, and the alpha Tolman. What's sort of interesting is the Reading and Ashbrook are clustering together, and down here we have Whitney, Tarentaise, and the alpha Tolman. And that kind of makes sense again if you think about the Ashbrook and Reading right here, Ashbrook and Reading right here. Those look very, very similar to each other. And actually, because they have a lot less of the Corynebacterium and other diversity in them, the Tarentaise, Whitney, and alpha Tolman are clustering more closely together to each other in this ordination space. One thing that we did in addition to the sequencing is we did a lot of plating, because the plating is easier to do, faster to do. And it's something, like I said, that some cheese makers can potentially do on site with relatively simple lab setups. And we did the plating particularly to look at harbison, And what we found is that the variation that we saw over time in Harbiston was reflected in the plates as well. So just to refresh your memory, the variation we saw over time in Harbiston, a lot of that variation is coming from this reddish kind of hot pink color here. So you see some of the Psychobacter, the Pseudoalteromonas and Halomonas and Vibrio here, and then it kind of goes away, and then it kind of comes back. And what's really great about those bacteria, they're called proteobacteria. And typically on plates, they make these large kind of beige, I guess is the color for this, or tan, light tan blobs on this particular medium that we use, which is called plant count agar with milk and salt. And those other bacteria that we were finding, those are including Actinobacteria and Firmicutes. They're typically quite colorful. They don't make as large of colonies, and they typically aren't as shiny. And so just like we could see over time, sort of blooms of proteobacteria switching to less proteobacteria, more actinobacteria, and then sort of switching back to higher amounts of proteobacteria. We could see that very simply in our plate. And this could be important for cheesemakers because it's known that proteobacteria and their growth in rinds, and a little bit in the paste even, can contribute cooked cabbage flavors, sometimes really strong sulfur flavors, and sometimes really kind of unpleasant flavors as well. And so in this case, we didn't really find any unpleasant flavors. These cheeses were totally delicious and not unusual. And you'll hear more from Roy about that. But it is interesting if you think about a microbial composition that looks like this, or even this versus this, what does that mean for the overall quality and aesthetics and flavor of the cheese? How is that shift potentially changing the way that the cheese tastes? And so that's something that I think we now see this variation, and it'll be important to be able to link that to data that Roy and Scott Frost have collected as part of this project to understand how these shifts in the microbiome can affect the overall both sensory perspectives, how the consumer perceives it, but also the specific chemicals these microbes are making. So what are some of the summaries of what we learned? Well, we've begun to establish baseline profiles for both the rind and the paste for these 6 types of cheeses, which represent sort of classic New England artisan cheeses. We've also identified that a lot of the variation is really in the rind and not the paste. And so that might be an important place to sort of focus efforts to be able to manage these cheeses. We've also demonstrated that there is a lot of variation in the rind and specifically in Harbiston. And what we're trying to do now is understand why, right? So if we go back to those data, why are we seeing such dramatic fluctuations? The recipes are the same, cheesemakers are doing the same things. What is actually causing these dramatic shifts? Another question is, does it matter? Maybe these cheeses actually don't taste that different, but have similar or have very different microbiomes. We don't know. Something we're trying to figure out. And once we can figure out why we're getting these big shifts, we might be able to figure out how to better manage these microbiomes. What is it happening in the cave? Or what is it about the microbes that are being added, or the amount of salts, or maybe even the time of year and just the microbes that happen to be around that could be affecting that variation. And so that's a summary of what we've been learning. I think all of this, all these approaches, all these techniques can be very useful for other cheesemakers in the industry. I tell people all the time that having baseline profiles for your cheeses is so important, because when things aren't working right, you can't know what you should have if you don't know what you historically have had on your cheeses. So if I take a sample of something that isn't what you want in terms of quality, I won't know what should be there because we won't have that kind of data. So having these baseline data, I think, are so incredibly important. We actually encountered some defects from these cheeses along the way, and those baseline data were able to help us figure out what was there that shouldn't be there, and then think about ways to manage that. So I think that's really important. And I also think thinking about both the paste and the rind is really important. I think the other thing that's been really important about this project is just figuring out how to collect microbiome data in the context of both sensory and chemistry data. Typically, this is all done in isolation, and integrating across these different approaches, I think, is really, really important. And with that, I would just quickly thank Roy, who is one of the leaders of this particular project, as well as various people that helped with sample collection and other things, especially Shayna Wagner, who's right here. She was the undergraduate who did a lot of the DNA extractions, and did a senior thesis on this particular project. She was a Tufts undergrad, and I'm really grateful for all the time and energy she put into this. Also like to thank the cheese makers at both sites that helped us get these samples and work with them, and other members of the Wolf Lab that were helpful. And also thanking Scott Frost, who did the chemical analyses and was a helpful partner in putting all this together. And with that, I'll stop this presentation. Thank you for your time for listening. And if you have any questions about this, you could always reach out to me. I'm Benjamin Wolf, benjamin.wolf@tufts.edu if you have any questions. Thank you very much.